Hello and welcome back to Beyond Networks, the evolution of living systems. In last lecture, we came sort of back to Earth and, tried to explore what can we do with the tools that we actually have right now within the Newtonian paradigm to sort of get a new or increased and enhanced process perspective on evolution. And I was sort of advertising different ways, practical ways to get appropriately formulated and validated models for dynamic mechanistic explanation, morphogenetic fields, through these sort of three approaches that we went over: the ensemble approach of modeling lots of different network models, the forward modeling approach, biophysical models, biochemical models that have carefully measured parameters, and then the reverse engineering approach, where you sort of infer the value of parameters that you cannot measure. So this gives you a sort of a snapshot of the process of evolution, a little exploration of the local configuration space as it is right now. And that's already a very sort of powerful enhancement of what we know about evolution, even if it cannot deal with whole organisms and all that. So today, we're going to look at how this can contribute at this moment to the theory of evolution by looking at the problem of evolvability, which will keep us busy in this lecture and also the next one in a way. So evolvability, as we've heard in the lecture about Evo-Devo, is a really central topic and a very broad topic. And there are different definitions of the term, which I have already introduced. Just to, as a quick reminder here, there is evolvability in the sense of traditional sort of evolutionary genetics, as formulated by David Houle here. It's the ability of a population to respond to selection. So it's sort of something that depends on the standing genetic variation, on the sort of heritability in the technical sense of the term. And this is not what we're going to look at here today. So we're going to sort of focus on two slightly different ways of defining the term. One is provided here by Günther Wagner, who defines evolvability as the genome's ability to produce adaptive variants. I would say the developmental system's ability to produce adaptive variants, but otherwise this definition is really good. It's a measure of the accessibility of the phenotypic neighborhood of the system, or more precisely, the ability to produce potentially adaptive variants. So it's the old problem of why doesn't my laptop improve if I randomly change it, right? But why do organisms sometimes have mutations with beneficial effects? The last sort of sense of the term, more radical, as defined by John Maynard Smith, it is the propensity to evolve novel structures. And we've been over that as well. This is sort of a difficult term. What is novel? I mean, we're going to go into this a little bit today. But Andreas Wagner basically suggested to rename it to innovability, to distinguish it from the sort of weaker definition of just the ability to produce adaptive variants, which don't have to be novel in the sense of Wagner. Okay, so these are the different ways in which the term is used. We'll focus on 2 and 3 here. But first, before we go into the topic, just a very brief historical excursion. Do you know where the term evolvability comes from? It was coined by none other than Richard Dawkins in a bizarre book chapter, a contribution to a book called Artificial Life in 1989. And so Richard Dawkins was sitting in front of his, rather ancient by now Macintosh, using a little programming environment for kids, really, originally designed for kids. It's called Logo, and it has a little turtle, and you give the turtle instructions on how to travel on the screen, and it draws patterns. So this is what Richard Dawkins did. He was drawing patterns, and he was selecting different rules that gave him different shapes of patterns. For example, he was selecting for airplanes, maybe here, for bugs here, or for sort of aliens, I don't know what, here. And so while he was playing around with his computer, he noticed a bunch of things that was, that were kind of unsettling to him. And so he starts the chapter with this remark, a title like The Evolution of Evolvability, which is the title of this chapter, ought to be anathema to a dyed-in-the-wool radical neo-Darwinian like me. Indeed, it should. Because evolvability requires not only replicators, but also development. That is some sort of genetic genotype-phenotype map. Okay, and that's what we've been going over in many of the lectures in this course, okay? Development is required for evolution. And he sees it here and then never again. Biomorphs implement development as recursive drawing rules, the logo programming language, and artificial selection is implemented by Richard Dawkins sitting in front of the screen, clicking for selecting different shapes. And what he notices while doing that is that there are what he calls evolutionary watersheds. These are rare mutations that lead to their carriers being better evolvors. That's a term he coins as well. An evolvor is a lineage that brings forth a lot of different shapes. Okay, more than others. So it's easier to select for different shapes in those lines. And then he's sort of puzzled because it's not quite clear why this doesn't involve species or organism level selection. He's just been clicking on critters. Okay, so it's sort of a secondary effect, but it's not quite clear how it works. And this is going to be the topic of this lecture. How does this work? How do you get the evolution of evolvability, which according to some should be sort of the central point, aim, the central subject of study of Evo-Devo, okay, as suggested by Hendriksen and colleagues in this paper. So this is a big topic, okay, and it's really useful if we want to investigate this topic to take a process perspective, because eventually evolvability depends on characteristics of the developmental system, which sort of results in the covariance of related traits, or sort of the flip side of that coin, modularity of the genotype-phenotype map. We'll talk about these two in the next lecture. It leads to developmental constraints or variational properties of a developmental system. It is the developmental system that produces the variability in the first place. So in this sense, it's not really constrained. And of course, developmental systems are canalized, robust, and this will be what we talk about today. So this sort of interplay between evolvability and robustness, this is an old topic. And also, even though Richard Dawkins coined the term evolvability, so the recognition of the problem goes further back. We could argue to Waddington, who explicitly or implicitly, more or less, has it in his epigenetic landscape, right? I mean, it's sort of the aim of the landscape to explain certain landscape shapes will be more conducive to evolution than others, but it's not really explicit in there yet. So the first one to really explicitly state the problem is probably, once again, Rupert Riedel, who's pioneered a lot of the topics in our field. And so he states that genomic activity can be described as a complex algebraic formula. He's sort of a formal thinker, right? And that's sort of also a premonition of the genotype-phenotype map that he's not really using, which changes during evolution. So this structure, this complex algebraic formula is evolving itself. This is why I put Rupert Riedel in with the process structuralists. It's very, so the research aims are very similar, although he never sort of belonged to any kind of school or anything, or the process structuralists weren't some sort of coherent school, really. He says systems become increasingly interconnected during evolution. The more interconnected a component of the system, the harder it will become to solve. And of course, this is his concept of burden that I've mentioned when we talked about theory in Evo-Devo, which Wimsatt calls generative entrenchment. So these are the sort of components that a lot depends on, so it's getting harder and harder to change them. And this is a very simple way in which different parts of a developmental system will be more or less evolvable, okay? So because each new innovation increases burden, so that the more ancient, more central parts of the system will be less evolvable. And that was Rupert Riedel's explanation of why there are such stable Baupläne body plans in the phyla after the Cambrian explosion. Evolution is accelerated along certain directions, but cannot change in others, basically, because some parts of the system are burdened and some are not. So you get, in the end, biased variation, which is the basis for evolvability. You need biased variation to get evolvability. So, but, okay, so we have this fact, this observation that developmental systems are very robust. So also early on, people started discussing this apparent paradox. Okay, so robustness can be defined as the invariance of phenotypes in the face of perturbation. All right, and this seems to go against sort of evolvability, doesn't it? the mathematical definition given here by René Thom of robustness is structural stability. And one really important point of his catastrophe theory was that these sort of topological catastrophes were very robust against details, changes in the details of the system. So a robust phenotype implies robust dynamics in the underlying system. Structural stability means that the qualitative behavior of trajectories is unaffected by small perturbations until something really bad, really big or bad, the catastrophe happens. Not necessarily bad. That's a bad connotation of catastrophe, but something radical happens, right? Okay, so this is a fundamental characteristic of evolvable developmental systems, because you have to be sort of stable to produce reliably every generation the same sort of form, right? And the perturbations you encounter are many. Okay, so you can have fluctuations in your environment, also regular seasonal changes, for example. You can have genetic mutations that affect different individuals in the population, and or stochastic fluctuations. Shit happens, lots of accidents happen. And you want to be robust against that. Okay, so as I said very early on, when talking about anti-fragility and Nassim Taleb's sort of ideas, what makes living systems different from others, evolving systems, evolution is sort of an anti-fragile system. It learns from those perturbations. Okay, but so all of this, how does this work together with the ability to produce adaptive variation? Okay, if you're robust. So how can you be evolvable if you're robust against mutation, for example? And here comes a really, really cool little paper by John Maynard Smith. So he's one of these examples. I disagree with his view, his ultra-reductionist view of evolution, but the man is full of really good ideas, and he was really open. He hired my master supervisor, Brian Goodwin, at Sussex at the time. Very open-minded, okay, allowing different perspectives in his department. And so he must have sat down somewhere for an afternoon and written this paper for Nature, which is 2 pages. It's wonderful. It's a real conceptual piece where he asks the questions: how does nature find functional phenotypes in a vanishingly small fraction of functional phenotypes? In his case, protein sequences in this absolutely humongous space of possible sequences. And that's a valid question. It's a real problem. I mean, if evolution can't find something, if it's not structurally stable enough to be found in the first place, it cannot evolve. And so he came up with this little word game that we already encountered when we talked about lineage explanation. And sort of this idea that pretty much any word in the English language of 4 or 5 letters can be turned, you can turn one of those words into any other through a series of functional steps. So here, word, or, gore, gone, gene, right? So, and he says protein evolution must work the same way. Functional proteins must form a continuous network which can be traversed using by unit mutational steps without passing through non-functional intermediates. Otherwise, you couldn't evolve. So these sort of networks of proteins are a prerequisite for evolution. And so this little thought experiment, great impact, massive impact, led by the study. So this was then formally studied by Peter Schuster and his group, Walter Fontana and other people here in Vienna, in RNA secondary structures. And that's the same thing. So you have these networks of different RNA sequences that form the same structure. They called this a neutral network because the evolution, the mutational changes didn't have any effect on the phenotype, okay? And so the same can be applied to gene regulatory networks. This is a beautiful pioneering study by Stefano Giliberti at the time in Andreas Wagner's group. And so what they did is they made some really simple mathematical network models. So there's a simplified version of the sort of the gap gene model that I was showing to you. And there's sort of a matrix of genetic interactions. And it's sort of an on-off model. So the genes are just 0 or 1. They're either on or off. And so these sort of regulatory weights, they filter the input and end up being above or below a sort of threshold. So you add up the values that are filtered through these weights, and the gene is either on or off, depending whether it's above or below a threshold, and so on and so forth. Okay, so you get a matrix of interactions where there's a number, a weight, just like in the gap gene model that represents every interaction between the different genes. So each gene has a row here, and all the regulators have their separate columns. Of course, they're the same, right? Genes are both regulated and regulators. So in this very simple model, you can just run the model a couple of times and see if it arrives at a sort of a steady state pattern. And then you can group these models into circuits that produce the same output pattern, right? And what you can also do is you can make a network of networks, which is really interesting. So you can mutate the networks. You can change one weight in the matrix and then sort of make this network of networks where the neighbors are just one mutational step from each other. So here, for example, this interaction has been eliminated. Here it's this one, and so on and so forth. So Andreas Wagner calls this network of networks a genotype network. And so you can actually simulate random networks like this and group them, and you find that these networks of networks, they form huge connected components in the space of all possible networks. Those that make the same phenotype, right? So what you see here is a graph of each dot is one of these networks, and they're all connected, and they all produce the same sort of phenotype in the end. Okay, so this is quite amazing. So the conclusions from this study were the following. So environmental and mutational robustness are closely correlated. It's not entirely surprising. So if it doesn't matter how you perturb the system, I mean, they had different ways of perturbing them by flipping the state of genes or by mutating the matrix, and they had very similar effects. That's not the main point here. The fraction, that's more important. The fraction of viable networks is very, very small. Okay, and the larger the network, the smaller the fraction. So it's vanishingly small for these sort of networks that we're looking at for developmental processes. And viable networks are generally connected in one single large component, a genotype meta network. And you see that the approach here is, of course, the ensemble approach. They just sample a whole lot of different random networks, yeah, to detect these sort of connected components. So a genotype network is an important concept. And the important thing to remember is that it's a network of networks. So it's like a meta network, networks connected by mutational steps. Robust networks tend to evolve from non-robust ones. Okay, so we had this problem with, so Dawkins was utterly puzzled by it, but also Riedel says, how does evolvability evolve? Okay, you cannot select, so maybe we should state this more explicitly. You cannot select for evolvability because it's a property of the lineage, not of an individual. And you know, especially in Dawkins' view, you should not be able to select on a lineage. This is group selection. Okay, you select on individuals only, and in those models, you only select on the individuals, but you get this lineage sort of level effect, which is really interesting. So how does it happen? It happens because non-robust networks fall off the genotype network in response to mutation. In this sort of simulation, the ones that fall off are not viable. Okay, this was already suggested by Van Nimwegen and colleagues in 1999 that this is a feature of network evolution, and it was just rediscovered here in this study. So basically, to show a picture of that, so the structure of these genotype networks is extremely important for the system to be evolvable at all. If you have low robustness, you get very sort of low innovation because there is no network that you can evolve on. Okay, so you imagine that there are different sort of new phenotypes that are desirable under certain selective conditions. One is here, one is here, one is here. But the evolution cannot happen because there is no sort of neutral network to traverse the space to get here, here, here. If you're here, you're stuck. On the other extreme, if you have really high robustness, too much robustness, you also cannot get anywhere, right? And so the discovery of these sort of really sort of rare but very, very large networks, they span a lot of the space of possibilities. They form sort of bridges between those different new phenotypes. But the most important thing is if you're at the outside of this network, you have a much higher probability of falling off. So the network will tend, a population of networks will tend to accumulate in evolution in here. And that increases the average robustness of the network. So you not only get the evolution of evolvability, you get the evolution of robustness, which are both sort of lineage systems level properties. Okay, so these are sort of secondary effects from just looking at the basic principles of network evolution. Really interesting in my opinion. So genotype networks span a large proportion of genotype space under all kinds of different assumptions on simulations that people have done. This has been confirmed. Neighboring genotype networks vary a lot depending on where in genotype space you are. Okay, so you can have different, you can access different alternative phenotypes if you're here on this connected network. And genotype networks allow you to traverse the vast distances in genotype space required to discover new phenotypes. So it's like a bridge between these new opportunities here. And this resolves the paradox of robustness and evolvability. Phenotypic evolvability requires robustness at the network level. So you have to sort of take it apart. Levels of organization, robustness at the phenotype is not the same as robustness at the network level. Okay, and if you properly take these apart, you understand why mutational robustness is a requirement for phenotypic evolvability. Beautiful work, all based on this sort of ensemble approach to network modeling. So this is looking at very general principles that if you assume some type of network, evolutionary network dynamics, the dynamics of the evolution of dynamic networks, you automatically get these second order effects of an evolution of robustness and evolvability, even though you have only individual selection. No miracles involved. Okay? To summarize this, once again, if you have lower robustness, the population just cannot spread very much. You know, these sort of, there's too many networks that fall off the neutral network, while if you have high robustness, you can spread out much faster and much further. And so you get a type of evolutionary dynamics that is described by these boom and bust cycles of diversity. So you spread out over a neutral network, and then when selection pressures change, you switch over to a new neutral network. And then you spread over that, and so on and so forth. Okay, so this explains punctuated equilibrium, for example. Non-gradualist evolution, and it also explains why so you have to have this robustness, because network evolution proceeds by neutral diversification along genotype networks. Network drift, which is a generalization of developmental system drift. Developmental systems are also some kind of networks, and those are interrupted by rapid selective sweeps for new adaptive phenotypes. Fantastic work by Andreas Wagner and colleagues over many, many years. Really good stuff. So this is another aspect of innovation, right? So it is a necessary ingredient for getting innovation in evolution. Robustness is a necessary ingredient, which is sort of surprising. But these are sort of all, you know, in silico simulations. What's happening in the real world? What is the, so for now we have a sort of a simulation-based explanation, but it's not really yet a causal mechanistic explanation. We do not know why this is happening. Why do these genotype networks exist? Okay, and the explanation lies in the particular configuration space of dynamical systems, okay? Because, so imagine you have a genotype net. So here I'm depicting a bunch of genotype networks. So a green phenotype, a blue phenotype, and a light blue azure phenotype. So you're sort of imagining that these are different states of gene expression, for example. And if you look at different networks in the node, they have different phase spaces. And if you look at how the geometry of phase space changes, I'm showing you a bunch of representative examples here. So we're always starting from the same initial condition, and we see, where does the system go if we're here? So if you have this sort of shape of phase space, you end up at the blue attractor. So this is on the blue genotype network, producing the blue phenotype. While here, there is no bifurcation that happened, but the separatrix has shifted, and you now end up in the green area. Okay, so this is a green in a green genotype network. And then here, if you cross this red line, that's a bifurcation boundary. Remember Albert's diagram? So Albert wasn't quite right. actually the changes in the phenotype don't necessarily happen at the bifurcation boundary, because you can have a bifurcation like here and still go to the same phenotype for a while. So these green dots still go to the same green phenotype until you actually fall into that basin of attraction and produce the azure third phenotype that has come into existence through this bifurcation. So the insight here is that two insights are very important. The underlying shape of the phase space and the parameter space, they are the cause of the presence of these genotype networks. And the other important insight here is that the picture is a bit more complicated than Alberg suggested, which is drawing these bifurcation diagrams and then saying, okay, these transitions can happen. Because there is often a delay. You cross the bifurcation boundary, but there is a delay in then switching the phenotype, because you have to first fall into that new basin of attraction that emerged through a bifurcation. Okay, so this is the theory. How does this work in reality? Can we study this, empirically? And so, sort of, what this is, of course, is a practical sort of example of developmental system drift. Okay, so the network drift that we're interested in Evo-Devo is this is happening in developmental networks. So this is basically, in the end, ultimately a mechanistic explanation of developmental systems drift. So this is work that my empirical research group did in Barcelona, again on the evolution not only of Drosophila here, but different sort of fruit flies. And just to give you a bit of evolutionary background, you should know that the segmentation process in flies is very derived. It's very strange. Okay, so these animals are developing very fast, and so most insects have this sort of very different type of segmentation process where they grow and they add the segments one by one. So sequential segmentation. The technical term is short germ band segmentation. While Drosophila is sort of subdividing an existing embryo without growth, and this is called long germ band segmentation. So this is very derived, and one of the big questions in the field is, how did this evolve from that, right? I mean, this looks like a huge transition, and so there's a problem with evolvability. How did this happen? How did one mode of segmentation transition into the other? And so we've done a bunch of comparative studies in different species of flies. So we looked not only in Drosophila at the maternal gradients shown here. This is bicoid, caudal, and hunchback, and these are the gap genes. Don't worry too much about them, but hunchback in yellow, crouple in green, knurps in red, giant in blue, and here are the terminal gap genes, talus and huckabine. And so we compared, based on other people's work already, we compared different species. In particular, we looked at this species, Megaselia abdita, which is called a scuttlefly, which is sort of interesting, because if you look at the phylogenetic tree of the Diptera, so the flies, and then over here the mosquitoes, there's a big divide, and there's this red group here called the Cyclorapha, or commonly called higher flies, unfortunately. I don't know what's more sophisticated about them than other flies. But these flies have a maternal bicoid gradient, and other flies do not. So one of the surprising insights that people had, Schmidt-Ott and other people who pioneered this sort of field, was that you had huge differences in the sort of gradients that go into the system. Okay, so the gap gene patterns in this red group, they look more or less the same in every species, but the sort of combination of maternal gradients that goes into the patterning are very different. This one is missing hoverfly, Episurphus balteatus, a very beautiful creature, is missing the maternal hunchback gradient. Megaselia is missing the maternal caudal gradient, and so you can have entire gradient systems missing and still get the same pattern. So what we're looking at within this group is, how does the system not evolve? Then when you move outside, there are differences in the arrangement of the gap domains and so on. But I'm not going to talk about this. So we're going to compare this fly, Megaselia abdita, with this fly, Drosophila melanogaster, and have a look at how the system evolved. And what we found is, we found two kinds of system drift here, okay? And I'm going to tell you this story right now. So the first type of drift happens between when you go from these maternal gradients into the gap genes. And because there's such drastic changes in expression pattern of these gradients, you need rather drastic changes in the wiring towards the gap genes. So this is a very complicated slide, but all you have to take from it is that if you look at these boxes here, they represent the maternal systems, and then down here, these boxes represent the gap genes. You can see that the wiring diagram between this level and this level has changed qualitatively. So you have added or subtracted new interactions, and this is called qualitative system drift. When you actually turn a repression into an activation, or you add or delete network interactions. So this system can evolve and react to huge variation in the inputs of the maternal gradients through system drift, because there must be some sort of underlying genotype network. And so we were trying to ask, what is that? What is going on there? And so for that, we moved to a more subtle sort of change in the expression pattern of the gap genes themselves. And what I'm showing you here, so what you need to remember is that these gap genes are shifting, right? So they're shifting towards the interior of the embryo, and they have this accordion-like movement. We've been over that, but quite a long time ago, so I'm just reminding you right now. And so what we're showing here are different peaks of gap domains, positions of gap domains in the different species. So in Megaselia, the solid lines, and in Drosophila with the dotted lines, and we're comparing them. And we found that the overall expression patterns of the gap genes, as I told you already, are very similar, but there are very interesting differences in the dynamics. So let me point out two of them that we're going to focus on. This one is interesting because the hunchback domain in Drosophila is overall static. It doesn't move, but in Megaselia it moves. Okay. So in Drosophila, a lot of people have sort of ascribed a very special role to this boundary in the overall patterning system, because it's the first stable boundary in sort of the middle of the embryo. Okay, while the more posterior and the anterior boundaries are more unstable, they move. Okay, and then here, very interesting. So in the posterior, you have a giant that comes up in Megaselia much further posterior, and then the shift in the domain you can see is much stronger, much more highly pronounced than Drosophila, while posterior hunchback comes up much later. So there's a general delay in posterior patterning because of there being no caudal gradient. So this posterior maternal gradient is missing that usually provides, in Drosophila, provides the activating input, these posterior genes. So there is zygotic caudal, but it takes more time to build up. And so the activation of these posterior gap domains is delayed, and so they have to shift quicker to sort of catch up with the rest compared to Drosophila. Oof, what am I telling you here? So I'm telling you that we're looking at very sort of subtle but interesting quantitative differences in gene expression. And these changes in gene expression are important here because, to go back to the evolutionary context, these shifts are sort of a dynamic fossil, a remnant of the original one-by-one addition mode of segmentation, which involves a sort of an oscillating gene expression. Okay, so these are very interesting features, and they're very labile. They change a lot from species to species, while the overall arrangement of gap domains is very consistent. So what we did is we did both modeling and perturbation approach to get to a dynamic mechanistic explanation of this phenomenon. Okay, so first of all, Carl Wotton, a brilliant postdoc in my lab, did this huge sort of assay with RNAi knockdowns of all the different genes, assessing quantitatively the effect on all the other gap genes. And RNAi has a usually nasty side effect, which is it's very sort of concentration dependent. So when you do an RNAi experiment, you get a lot of difference in the sort of response. It's very variable, and usually this is a problem if you're doing normal genetics. But we are very excited about this because you can assess, by sort of measuring a lot of the effect in a lot of different embryos. You can assess the severity, the strength of the interaction, not just the quality of the interaction through this assay. And so what we did is we reconstructed the gap gene network in Megaselia where nobody had looked before. And after 5 years of hard work by Carl and other people in the lab, we found that the gap gene, the qualitative structure of the gap gene network in Megaselia is exactly the same as in Drosophila. But because we did this quantitatively, we could tell that there were certain interactions in the system that differed in strength between the two different species. So we had done the decomposition in Megaselia, and now we did the recomposition. So we took this sort of reverse engineering approach that I introduced to in Drosophila already. We looked at the expression pattern in different species, and we quantified, or at least got some semi-quantitative data out of these different non-model organisms. Are very hard to work with, and so we needed a sort of a more improvised approach than in Drosophila. But we got a sort of a semi-quantitative data set tracking the expression patterns of those gap genes over space and over time, and we fit this gene circuit model, very similar again to the models that Andreas Wagner used for his ensemble approach, to these data sets to get suggested proposed network structures, and also, of course, phase spaces that we could analyze in these different species. Again, I'm only going to talk about Megaselia and Drosophila, but we also did it for this non-cycloraphan fly, Clognia albipunctata. So the idea here is we take this dynamic mechanistic explanation idea for a comparative approach, right? So what we found is that the models were able to reproduce these subtle differences between Megaselia and Drosophila. So what you see here are sort of the final patterns in Megaselia on the left and Drosophila on the right. The colored areas are the data we fit into, and the lines here are individual simulations that were fit to data. And you can see that they reproduce the two major differences very nicely. So there's a shifting domain of hunchback, which doesn't shift in Drosophila. And there is this sort of really weird behavior of posterior giant, which does not happen in Drosophila. So we wanted to explain those differences in these very subtle gene expression features. And remember, the subtlety of the features is just a sort of a proof that the quality of the data is very good. Subtle differences do not mean unimportant differences, okay? These differences are very important because at the end, both flies need to arrive at gap domains that are placed at exactly the same spots. Otherwise, you don't get a viable fly. Okay, so to sum everything up, what we found is that these differences, the qualitative systems drift in Megaselia had created a system with different shifts in the boundaries, okay? And those different shifts in the boundaries were exactly the mechanism that compensated for A, the missing posterior caudal domain, which leads to this, delayed expression of posterior gap domains, hunchback and giant here, okay? So the differences in these sort of interactions among the gap genes could account for the differences in the shifts, which accounted for the compensation for the missing maternal caudal. The same thing in the anterior, this shifting hunchback domain is there because the bicoid gradient in Megaselia is much broader than in Drosophila. Again, the whole pattern is shifted to the posterior, and so it needs to shift to end up, in the end, at the right position, which is identical in both species. So we thought it was really interesting. So first of all, we found a new way, a quantitative way of system drift. So what's changing here is not the sort of the sign or the number and the nature of the interactions, but just their strength, okay? And this allows you to fine-tune the reaction of the system, compensatory evolution towards changes in the maternal inputs. So this is a new kind of system drift and a mechanistic, causal mechanistic explanation of an evolutionary phenomenon. It tells you why there is a genotype network, and because of the underlying structure of the phase space. And it also tells you why that is important to get developmental system drift in the first place. So this is the first time anyone has ever done this. And it's also, so far, the last time anyone has ever done this. This paper came out in 2016. Okay, so it's not ancient history yet, but okay, we should be doing more of this, right? So what we're looking at here is very explicitly at the properties of developmental processes and how they influence the evolvability of systems, because we have now a mechanistic explanation of why certain features of the system are very labile to evolutionary change, the shifts themselves, and others are very robust towards mutational change, the sort of generic arrangement of these gap domains in the system. Okay, and so that is one part of this story. The other part is, of course, how do we map? I'm talking about mechanisms. What are these mechanisms exactly? Okay, I haven't told you much about this yet. So we will look, in the next lecture, we will turn our attention back to sort of the structure of the gap gene system and see what features in the structure of the gap genes are actually causing this difference. And with that, I thank you for listening, and I hope you'll tune in to what is really part 2 of this lecture the next time. Thank you very much. Bye now.